Welcome to the PESI presentation for Chapter 8, Section 5, Translation. What is translation? Translation is a process that converts or translates an mRNA message into a polypeptide. One or more polypeptides make up a protein. Codon is a 3-nucleotide sequence that codes for an amino acid. A, G, C, and T for DNA, and A, G, C, and U for RNA are the types of nucleotides. For example, the segment of mRNA is expressed in A, G, C, and U, whereas if this was a segment of DNA, then it would be expressed as A, G, C, and T, not U. Many amino acids are coded by one or more codon. Amino acid leucine, represented by 6 different codons, CUU, CUC, CUA, and so on. The first 2 nucleotides are generally the most important, for in this case CU would be the 2 nucleotides, in coding for an amino acid. This feature, CU in this case, makes DNA more liberal of many point mutations. The 2 major types of codons would be 3 stop codons, which signal the end of the amino acid chain, which will appear in the diagram later on, and start codon, which signals the start of translation in the amino acid methionine. Translation always begins with methionine. For the mRNA code to be translated correctly, codons must be read in the right order. Codons are read without spaces as a series of 3 non-overlapping nucleotides. The order of reading codons is called the reading frame. If the reading frame is altered, then the resulting protein changes greatly. The genetic code is known as a common language because it is shared by almost all organisms. Almost all organisms follows the codon UUU codes. This code is often called universal. The common nature of the genetic codes allows us to draw a conclusion that almost all organisms arose from a common ancestor. Ribosomes. Ribosomes are made up of a combination of rRNA and proteins. Ribosomes have a large and a small subunit that fit together and pull the mRNA strand through. tRNA acts as a sort of adapter between mRNA and amino acids. Cells need tRNA to carry free-floating amino acids from the cytoplasm to the ribosome. Anticodon is a set of 3 nucleotides that is complementary to an mRNA codon. For example, anticodon CCC equals mRNA codon GGG, but it can be expressed in 2 ways. Translation has many steps and takes a lot of energy from a cell. Translation takes place in the cytoplasm for both prokaryotic and eukaryotic cells. Small ribosomal subunit must bind to an mRNA strand in the cytoplasm in order for translation to take place. Well, here is a very careless screenshot from the textbook. Well, I'll go on with describing with the diagram. These are the steps of translation. The first step would be the exposed codon in the first site attracts a complementary tRNA, bearing an amino acid. The tRNA anticodon pairs with the mRNA codon, bringing it very close to the other tRNA molecule. See, the translations must start with methionine. And these 3 are the codons that signal the start of the translation. The ribosome forms a peptide bond between the 2 amino acids, and it breaks the bond between the first tRNA and its amino acid. The ribosome pulls the mRNA strand to the length of 1 codon. It shifts length of 1 codon. The first tRNA is shifted into the exit site, where it leaves the ribosome and returns to the cytoplasm to recharge. The first site is again empty, exposing the next mRNA codon. So each site is replaced, like every time. And the ribosome continues to translate the mRNA strand until it reaches a stop codon. Then it releases the new protein and disassembles. See, here are the stop codons, the 3 codons that signal the end of translation. And the main points would be obviously the complicated 3 steps of translation, and the fact that translation converts an mRNA script into a polypeptide. This process consists of 3 repeating steps. Anticodon is a set of 3 nucleotides that is complementary to an mRNA codon. And that's pretty much it for Chapter 8, Section 5, Translation. Thank you.